I'll be talking about the use of circulating tumor DNA to look at cancer genomics and how it changes over time, and the different research as well as possible clinical applications for circulating tumor DNA. The amazing fact is that if you take a blood sample and you get rid of the cells, you find thousands of copies of the genome present in every cc of plasma. And within this material, you also find DNA that comes from the tumor and different tumor lesions in the body. Analysis of this DNA with the appropriate methods allows us to get information about the genomic changes that occur in the tumor lesions themselves. So one of the things we're finding out is that by analysis of circulating tumor DNA in the plasma, we get a picture that averages out, if you will, the different lesions that are present in the body. So we see higher prevalence in the plasma of mutations that are present across the different lesions in the body, whereas some of the private mutations that are present only in individual lesions, we may miss, or we may find them if these lesions are particularly large or aggressive. One of the attractive features of circulating tumor DNA is that we obtain all this information from a plasma sample. And a plasma sample is minimally invasive. You can obtain it multiple times during the patient's course of treatment, and this allows us to obtain time courses. Initially, these time courses were only of individual mutations that we could analyze, but more and more we can now use the same material to look at large, extensive profiles. And now this allows us to get extensive profiles and how these change over time. The way we manage to get the information out of the liquid biopsies, if you will, is by integrating technologies that have been maturing and in existence, by improving them and integrating them in a way that is more effective. So the separation of the plasma from the blood is not new. The extraction of DNA from plasma is something that's been around for many years. Taking this DNA and using this to generate libraries from next generation sequencing, and then applying the right methods for that and the right methods of analysis allows us to really drill deeper and deeper in terms of sensitivity and also in terms of breadth of analysis. The information is there, and it's definitely doable and attainable, but it does require expertise and a certain degree of dedication to the task, if you will. So I think the best ways to access that are either through collaborating with academic labs that specialize in this, or more and more it's now becoming available as commercial services through centers or companies that specialize in providing this type of service. I should add that I'm personally a co-founder of one of these companies, so I have a conflict of interest in this. What we hope is that the accessibility, the ease of collection of these samples would mean that it would be possible to obtain the types of genomic analyses for more and more patients and multiple time points, and in a way to make genomic analysis more and more accessible to patients. Data is still accumulating on the to really delineate and define the clinical validity and clinical utility of these tests. Of course, in order for this to be commercially available, it needs to be set at the right price points and reimbursements and all these systems. But in terms of accessibility, I think it's coming fast.